Okay, we are online on YouTube. Camille, remember that we have 30, 40 seconds of delay on YouTube. So it's normal that the people, the public, will ask you questions with a little bit of delay, but it's normal. We do it for to have a better connection.
Okay.
Okay, we have 17 participants, so we can wait a couple of minutes and then you can start, if you agree.
Okay. Hello.
Good morning.
Good morning.
Hello.
Good morning. Your account was hacking by Filippo. Was hacked by Filippo.
Maybe that's why I had so many problems yesterday.
Yeah, because you did what she's used to.
No, but it's possible. Eh? I don't know how it's possible, but for sure. Okay.
Things related to the life.
Before to start, I give the permission to all the trainees to talk. Remember to close the microphone.
Before we start, maybe I would like to mention some things regarding the analysis on air, because we had a couple of emails yesterday afternoon.
Mm-hmm.
Give me just 1 second. I don't know why I cannot see the links. Can you send me the link on YouTube for today? I have it here, Thursday, 3rd of December. Okay. Okay, here we go. Is everybody already connected?
We have 20 people connected and 25 people on YouTube.
Okay. Okay, so I guess if it's all the trainees already here, good morning. Welcome to the third day of this Method training school. Today we're going to have the great talk of Camille and Amandine. So briefly, I would like to mention before we start that regarding the analysis that you will do for the with the AIR platform on the Sequentia website, basically you need to have 6 samples. So I know that many people have more than 6 samples. You can upload them and validate them, all of the samples that you want, but you will only have credit to analyze 6 samples. So if these 6 samples come from single-end experiments, single-end reads, you will have 6 files. If you are working with pair-end reads, you will have 12 files, okay? So the combination of samples is up to you. The only requirement is that you need to have at least 2 biological replicates. Otherwise, you cannot start the analysis. I know that many people have already started, and some of them have already done the analysis. So the idea is for those who have not yet started, if you have any doubts, to contact me during the day, and I will try to help you to either upload the data or validate it or start the analysis. Okay. I think that's it. Let's start now with the plenary session of today. So we are really excited to have Camila and Amandine with us today. They actually were also last version of the training school. They were also giving a talk, although remotely, regarding the Great Expression Atlas. And now both of them will try to give the basics of how this platform was developed, and then later on the afternoon and tomorrow afternoon will be the practical or training session for the use of this platform. So Camille Rostenholz is an associate professor at the Strasbourg University in France, and she's doing her research in the Colmar INRAE Institute since 2012, working in grapevine genomics. She leads a group there, and she's focusing in the characterizations of gene families involving aromas and resistance to pathogens. She's also related now to one of the latest resequencing of the Pinot Noir genome. So we expect to have real soon this new available genome data using the PacBio, right? Also, Amandine is a bioinformatician in Camille's group in RAE since 2016, and she's an expert in transcriptomics, genome assembly, and different tools and pipelines for analysis of omics data. So up to you. Thanks, Camila and Amandine, for being here.
Thank you, Tom, for the nice introduction. And I'd like to thank especially Tom, Jérôme, Anne-Marie, also Mario, and all the staff. You are doing really great. I mean, if I may abuse of this term, sorry, I'm going to this morning and this afternoon probably. But really, you are a great team, and it's a great pleasure to work with you and to spend this time with you for this training session of the COST. So I'd like to thank Amandine also, because I don't really even remember how I worked before she was in the lab, and she is doing pretty much all the work about the reference genome, as Tom just said, and also on the great so the Grape Expression Atlas that I'm going to present you. So maybe some of you have already heard about this presentation, but I'd like to introduce it anyway. And and present you the philosophy that is behind this tool. And I really like to show you that GREAT is not only a web interface, but it's also a curated database, an analysis workflow that we are actually sharing, and a web application to analyze RNA-seq data. Before starting, can you see my slides, my pointer moving?
Yeah, yeah.
Yes, we
Good.
can.
So
Perfectly,
I move on with that. So hang on. So GREAT was born about, let's say, I don't even remember really, but it was about 6 years ago. When I was just freshly hired in the lab, and my colleagues, my boss Philippe, came and see me and say, "Hey, would it be possible to look at the expression of my favorite genes using all the RNA-seq data sets available?" Just that. And also because he's so fond of heat maps, he wanted me to draw nice heat maps all around. So okay, that was the first ask. I mean, the first time that I was asked for such a tool. And then another day, Eric, maybe you know Eric Duchesne in the lab. So same, he came and see me and say, "Hey, could you help me to look at the expression profile of my genes within a QTL interval, because I want to find candidate genes?" Okay. So he wanted basically to look at the expression profiles of the genes for every conditions, every organ, every developmental stages of anything, everything, just to have a clue of what his candidate gene could be in the QTL interval. So they kept coming and asking me about these things, and I was really thinking it was time to actually develop something. There was really a need to extract and translate for biologists, translate information contained in raw RNA-seq data that are stored in public databases. And because I thought, and they thought also, my colleagues, that this data could actually help them to answer their questions. And so at the beginning, I was actually performing the analysis, sending them the results. And then I got a little bored, and I thought we really need a tool, something that they could use on their own to look at expression of genes, any genes that they wanted to look at. So that's how everything started. But at that time, there were a lot of gene expression interfaces already that were or became available in between. So what I want to say is that all these platforms, all these web interfaces are really good, and we do not want to compete with our GREAT tool. We don't want to compete with them. I think that really in the omic world, diversity is really a strength. And I want really to show that if we can have a lot of very diverse tools, it will help biologists to answer their questions in all kind of ways. There are all kind of biologists. Maybe they feel better using Vespucci or another browser because they understand the philosophy behind better. They may have some questions that the GREAT platform cannot answer. So really what I want to say here is that all these interfaces, all these tools, and with the GREAT tool as well, all of these are complementary, and we are not competing with each other. So Vespucci is very good because it actually allows to analyze microarray. data as well, along with RNA-seq. So it's a very exhaustive tool. The thing is that using microarray, you have always to use samples to perform comparison, which can sometimes trigger some problems. Let's say if you have some specific biological questions, but Vespucci is very good for gene networks. It's very good for functional gene annotation, and really, I think that this tool is very good. The GRAPE RNA interface, so is developed by Chinese people, and I want to take my notes here. So yeah, it's very exhaustive as well. So you can perform blasts, you can look at transcriptome that they assembled. You can also design primers on these. It's very exhaustive, very full of tools. This web interface. The only thing is, in my opinion, it's not that very intuitive. It's not easy actually to move from one tool to the other. But really, I think there is nice stuff to do with this platform as well. VTCDB is a database that is based on co-expression, so which is a very nice idea to look also at genes that are co-expressed. It's based on microarrays, but unfortunately it's not maintained since 2014. So that's another problem, is that sometimes you have nice web tools, but they are not always maintained. So we need to actually keep developing tools to make the most of the new expression data that are coming up. The GRAPE EFE browser, I really like this interface, because it's I should have taken a screenshot of this, because it's nice. It's actually based on the drawings of the different organs of the plant. So the leaves, the berries, the different tissues, for example, the skin, the seeds, and showing with different shades of colors the level of expression for a given gene within these different organs. So it's the same kind of representation that you can find for Arabidopsis or tomato. Really nice one. But again, this one is not really maintained, because you cannot find the new expression data sets that are analyzed in this interface. And then there is another one, which is GDB, which is mostly based on resequencing data, so not expression data. But there is also a part of this interface where you can get a clue of the expression of the gene that you're actually looking at. So really my point here is that all these tools, and the GRAPE one as well, they have strengths, they have, I'd say, limits or weakness, but all of these are complementary and should be useful for the GRAPE community to actually keep going with the omics data. So that's really a take-home message that I want you to get to this morning. So it's not only about GRAPE, but it's about complementarity of the tools. So now let's focus on GRAPE especially. So GRAPE is meant to be a data mining tool, and it's not, and I want to stress that out, it's not a tool to perform differential expression analysis. So it's really different compared to to the air platform, for example. So it's really meant to perform data mining, and currently it's mostly used by biologists to run some preliminary analysis to check if their genes are expressed in one or another conditions. But it's really a tool to explore and to look into the data. So it's meant to make full use of publicly available Vitis vinifera RNA-seq data. So it's a tool that is only showing, analyzing Vitis vinifera RNA-seq, not any other Vitis species, because we wanted because as we are using actually the reference genome to assess and to measure gene expression, we were afraid that if we use other expression data set from other Vitis species, the genetic distance between these species would cause problems to actually make gene expression comparable. I don't know if it's very clear, but really we wanted to stick with the Vitis vinifera because we were pretty sure and confident that then the gene expression that we were comparing and looking at were actually comparable. Then, so we are using, that's a very important point, we are using the exact same normalization across all the data sets. So which it's based on, I'm going to explain that later on, it's based on an RPKM-like normalization, and that helps to actually compare every gene with each other in every sample that you want to look at. So there is no reference gene, there is no reference data set. And so really what we wanted with the GREAT is actually to be able to compare every expression value, so the RPKM-like value across all the samples and all the genes. So that's really the thing. And so it's very different, a different philosophy than performing a differential expression analysis. We also wanted GREAT to allow the viewing and exploration of expression profile of genes within a given genomic regions. So to help my colleague Eric Duchesne to look into his QTL regions of the genome, and all the others, not only Eric, of course. We wanted to be up to date with the genome assembly and gene annotation versions. So we really want to use the latest versions for reference genome and gene annotation, and you will see that we also want to help the users to actually find to be able to find back their gene ID across the different version of gene annotation, which is not an easy task. So we'll see that later on. And the point here is that we really wanted to be fair. So GREAT is actually a tool that is using the fairness of the data as we are actually reusing, reanalyzing publicly available data sets. But we also want to be fair, and in this way we are actually sharing all the database that is behind the GREAT tool. We are sharing all the pipelines, the workflows to perform the analysis of the data, so that, and also the scripts to actually actually show the web interface. So basically, what we wanted is that basically any bioinformatician could actually take GREAT and use it to perform the exact same thing on any other RNA-seq data that he wants. So make it great for, I don't know, Apple, great for any kind of species that anyone wants. So we really wanted to be an example of fairness using fair data. So it's yeah. So we really wanted to be fair. So how does it work? Great. So I told you it's a database, it's a workflow, and it's a web application. So I'm going to present you these three parts. So as I said, GREAT is a curated database for Grapevine RNA-seq data. So there is a huge amount of work in this only single little word, curated. We'll explain you that. So then we'll briefly go through the workflow for the RNA-seq data analysis. And I thank Ricardo because he introduced so well the RNA-seq analysis workflow yesterday. So I won't go into much detail today. And then we'll have a look at the web application to explore actually the data in the GREAT platform. So let's start with the curated database. So we are starting with the SRA database. So as it was explained, when was that? Tuesday, maybe in the morning. So the SRA database is a database that is actually storing raw data of all kinds. So it's meant to be a sequence read archive. And so, yes, it's storing raw data. It's a public repository for raw sequencing data. And it's where you actually store and where the sequence are submitted after a publication. The SRA stores actually the sequences in its own format, the .sra format, but it's very easy to convert into a FASTQ that we can and we can analyze this format very easily. And also the SRA database provide all kind of all the metadata that were actually submitted along with the sequences. And that's where the curated little word starts. And it's a huge amount of work. So it's a colleague of mine, so in my group, Lauriane Renaud, who is doing that. So every 6 months or so, she goes back to the SRA database, and she's doing this request. So Vitis vinifera RNA-seq, and she's then looking at all the new RNA-seq data sets on Vitis vinifera in the SRA database. And so she retrieves, she actually compared that with the accession number that we have already. And then she looks into all the metadata that are submitted along with the sequences. And so she takes all kind of information related to the raw data. So it's stored, these metadata are stored like in this kind of web pages, but you've seen that already with Jérôme and Tom, so I won't stay long here. So basically what she does, Lauriane, she actually collects metadata from this web page, and sometimes she could not get all the information she actually needed, and she most of the time needs to go back to the publication to actually fill the gaps in the metadata stored on that web page. I can see conversation.
Camille, can I
Yes.
ask a short question?
Yeah, sure.
So there's a lot of data in this repository that has not been published. In the case that data is not well annotated, what have you done?
We cannot do much. We do the best we can. So basically, we deal with what we have in hand. So if it's not, if the metadata are not correctly annotated or submitted, sometimes if it's too little information, we won't use the sample at all, because it's impossible to actually know exactly what it is. So it happened that we actually did not use some samples because we didn't have enough metadata associated with that. So yeah, does it answer your question? So it's really important, and that's one of my other take-home messages. The submission of the data is good, but if you don't have very good metadata associated with that, it's almost useless. So we can see that every day with the great platform here. So really, it's very important. I know it looks complicated, but I know that Jerome and Tom did a wonderful job at explaining that to you yesterday and the day before. It can look complicated and boring and everything, but really it's a need if we want to make good science again and again using this data. It's very important.
Yeah, let's hope that it's the take-home message of all the training school.
Yes, and for the observers too. I hope so. Okay, so yes, just do not hesitate, Tom, Jerome, Amandine, or if you see that there are some questions that I can answer right now. It's I've prepared one step where we can make a break or questions, but
Camille,
yes.
Maybe now, because you're going to a little bit more in your presentation, but I was wondering if you can speak a minute or two about what we are doing in the cost on the part where you presented all the database, the different database, and at least mentioning that it's a very big effort that some people in the cost are doing, not especially me. So that's why I'm not the best one to speak about it, but to maintain a repository and having a list of all the tools that have been developed in Grapevine. Before letting you guys speak, also I want to mention something that I consider very important in relation to what you said about not competing between tools. So there is always some kind of problems, and you talked a little bit about it. It's that at some point these tools are not maintained. So people work on a project, they do it. So if somebody take over, this is not really a problem. Like this, it's allow us to have a constant amount of new information.
Yes, really. I totally agree. With,
yeah. But no, I just want also to mention that in bad cost action, maintaining a repository and providing the address of all of these tools that's something that we are we are planning to develop.
Mhm. Yes.
Also, I'm pretty sure that new tools or platforms will be developed probably by other groups in the future, but the idea is to have like a hub, whether it's in the International Great Genome Program website or in the Cost Action or a centralized place. It's good to have all of them integrated, right? Because they are complementary.
Yeah.
Yes,
I also
definitely.
agree that, yes.
And also what I wanted to say is that I only selected some of them. They are focused on gene expression, but there are many, many others. And you can do data mining or analysis of any kind of omic data using very different tools and complementary platforms. So this was just a subset related to what I was talking about today, but there are many others.
Yeah. Hopefully we have public repositories and analysis tools and platforms for other omics, right? Like proteomics and metabolomics.
Mm-hmm.
Yeah.
Definitely. Good.
Thank you, Camille.
Thank you, guys. Okay, so let's keep going with the GREAT, and especially about the curated database. So the way we start is actually, Lauriane, the colleague of mine, she's actually building the database. So she selects the data sets that that have enough metadata where the data set that fits actually the requirements of GREAT. So for example, they have to be from Vitis vinifera, not another Vitis species, for example. So she performs a kind of selection, and then the GREAT actually retrieve the raw data sets from the SRA database to run the workflow on our calculation server at INRA Colmar. So the file that Lauriane is creating, and she keeps updating, I should say, it's an Excel spreadsheet, so an Excel file. And so every 6 months, she collects a new information. So what kind of information does she collect? So we can split this information into 4 categories, I should say. So first is really about who submitted the data, the name of the number of the bio project, the first author of the publication, or the first author of the person who actually submitted the data, a paper if there is any, the name of the file, so SRR blah 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 dot FASTQ. So we can actually link the name of the files with the name of the person who submitted the data. Then the second kind of information, it's related to really the sequencing of the data. The sequencing platform, it's almost only Illumina. If it's paired end or single end, the read length, the protocol for the library, is it a poly-A selection? Is it Ribose 0? Is it stranded or not? So these are information that are actually needed to run the analysis workflow later on. The third category of information is really related to the sample, and so that's where it's probably it's the most difficult actually to collect all these data. So it's about those species is usually easy. So we are only working with Vitis vinifera. Variety, it depends. It's not always mentioned which organ, which tissue of tissue of this organ. So, for example, organ would be berry, but in the berries you have the skin, you have the flesh, you have the seeds. So it can be, you can perform RNA-seq on all these different tissues. Then very tricky one is the development, the stage, the developmental stage detail. So it's not always easy to compare all the different stages that are mentioned in the metadata. Sometimes, so it's not always different developmental stages that are tested in the for the RNA-seq experiment. Sometimes a treatment was applied. So, for example, the biologist can perform biotic stresses on the plant. So, using a pathogen, or I don't know. So there are very, very diverse treatment that are applied on the plant. Can be abiotic stress. So we need to know what kind of treatment it is, the nature of the treatment. So treatment type would be biotic or abiotic. Treatment nature would be what kind of pathogen, for example. Is it Plasmopara viticola? Is it another one? So yeah, and then we need also details. Let's say it could be how many days after inoculation were the samples collected. So all this kind of information we are needed for to implement the database. And really that's where the big effort is needed, because it's sometimes very poorly mentioned in the metadata, and so Lauriane has to go to the publication and find the details and fill in this file. And the last category for the criteria are really for us. It's more like comments. For example, it happens we have a lot of information regarding this different treatment or organ or tissue. So we keep all the information in this comment part. And then we also have the quality check. It's when we actually run the workflow. Sometimes we get some samples that don't hit the specification, the threshold that we set regarding, for example, mapping quality or number of reads mapped, or reads that are actually assigned to an annotated gene, or this kind of stuff. So at some point we can also filter out some samples because the quality of the alignments or the counting is not okay. So just to give you an idea, right now in this database we have 1,991 samples, and they are actually, you can actually access them and view them on the web interface right now. So it's yet, because as you understood, it's a never-ending process, because every 6 months we get a huge amount of new data. So it keeps going all the time. So to give you an idea of what's in the GREAT database, so here, so Amandine did a very nice word cloud on the varieties that we have in the GREAT database, and the size of the variety name is related to the number of samples that we have in the GREAT database. So we mostly have samples from Cabernet Sauvignon, a lot of Pinot Noir, Merlot, but also Sauvignon Blanc, Chardonnay, a lot of Italian varieties too. I mean, great diversity of variety, but it's mostly still a huge proportion of Cabernet Sauvignon.
Camille?
A huge proportion. It's not that many. It's about 15% or 20% of the samples from Cabernet Sauvignon.
Camille?
Yes?
Is this only at the level of samples or also experiments, like complete experiments?
It's at the level of samples, really. It's not so it's 1,900, about 2,000 samples, really. If an experiment has control and then 2 hours post-inoculation, 3 hours post-inoculation, that would be 4. And every time you have a replicate, it's also a sample.
Yes.
So yes, let's compare it to what the trainees are going to perform tomorrow morning. So it would be 6 samples. What you'll be analyzing tomorrow, it would be 6 samples for the GREAT database.
Great.
Mm-hmm. Okay. About the different organs that we are looking at, so we mostly have berries and leaves. We decided to split the berries in at least 2. The green berries and the ripening berries, a lot of leaves, and also some flowers, inflorescence, but really berries and leaves make the almost the whole of the GREAT database right now. About treatment, so there's a huge diversity of treatment that are applied on our poor plants. It's for the good, of course. So different kind of biotic stresses. So we can see Plasmopara viticola, Erysife, Necator. We'll see this afternoon and tomorrow Neophysicocum parvum, and also a lot of abiotic stresses, water deficit, heat, what else, wound. Yes, trimming, all kind of different kind of stress that are under study. And finally, I'd like to show you also the different developmental stages that we have in the GREAT database, and that's where the problems actually are very important, because you can see that we have stages that are reported in different units, let's say. So it's BBCH, it's day after veraison, it's day, what do I have? Day after flowering, day. At least we were able to manage day after flowering and day after anthesis. We actually we deal with that, of course, but there are all kinds of different. There are the breaks also. I mean, it makes the comparisons very, very, very difficult, or almost impossible sometimes, because we just don't know what is really comparable with each other. So we are working a lot with Eric Duchesne and all the working group working on these ontologies, because we definitely need standardization here, or it's going to be even more problematic the more samples we use for the GREAT database, because we don't really know what is comparable with each other. So I think that the cost initiated here mean, it's probably it was in everybody's mind for a long time, but the work on standardization of the terms on the ontology used for developmental stages is crucial for the kind of database we're actually running. So if you have comments about that, or this is one also of my take-home messages, we need standardization to keep going with this kind of databases. And I know that the Vespucci guys, they are struggling with the exact same problems.
Yeah, I think that in Vespucci they are using the Eichhorn-Lorentz.
Yeah, we've got that too.
Yeah.
But the conversion is not easy. I mean, so right now we converted like when it was very easy, but we are really expecting waiting for the results of the cost, the working group about that, so we can actually simplify and use one common standard set of ontologies for that.
I think it would be always difficult to use one common standard, but at least to have synonym. And when we talk in BBCH or EL, even if it's different, we know exactly that they are related to each other.
Yes.
Paola said, is there any good example in the plant community? Maybe other plant example
for such kind of standardization?
Yes.
That's a good question. I don't know actually. Jérôme, maybe you know.
Oh, it's too. In that case, Grapevine is way too different to anything else. Especially, like you say, there is a bricks. Well, in other fruits, maybe people are using also the bricks. Yeah, it's true. But no, I think we are the people in the Grapevine community are the one doing the best job.
But also because it's probably an absolute need, because we have so many different ways to call a stage. I'm not sure that in any other plants it's getting so difficult. I mean, so diverse in the units we are using. I'm not sure. So Paola, I'm not sure we could answer your question properly. But anyway, I actually decided to make a question break here. So feel free to ask questions. Nope. Any questions on the YouTube?
No questions so far on YouTube.
Wait, there is one more question from Paola. She was just answering to your last comment, but it would be nice to explore if there is. But no, Paola, really, I answer to you. I think we are. Yeah, it's far from being perfect in the Grapevine for the Grapevine, but we are the one doing the best job, in my opinion. And now that I'm working in other fruit trees, I know that people are using day after flowering, day after anthesis. There is not so much an equivalent. I think the BBCH is a standard. Another way to do that also, yes, is to use the plant ontology that have some similarity to this kind of scale.
I see that Luciano has a question on the Q&A stream there.
Yes.
So maybe, so the question is, what's the link to the GREAT site? So I think that Amandine, you can copy paste the
Yes, I give the link, but
it's maybe not for the
an ID, so maybe I can send also an ID to connect.
Yes, Luciano, you need a login info to be able to connect.
Amandine, would it be okay if I send you all the mails of all the trainees, so or they need to register their own?
No, actually, the plan is for this afternoon. So we set up like 10 accounts for the trainees, and we'll give each trainee a specific login.
Okay.
And then if anyone is interested, and I should have put the email address here. So if anyone is interested in keep using the great platform, which I wish, so they just have to email us, and we'll set up a proper login account for them. And it's also true for the observers, of course.
Yeah, there is one comment on the YouTube channel, Stefania Pilati from FEM. She's been working in the Vespucci platform and second version. So she's commenting on the ontologies and the synonyms between plant organs and so on. So she says, "Dear Camille, it's a mess. We know."
Good. We are not alone.
Yeah. And she says, "We are trying to use the available ontologies on OLS and trying to convert when possible." So Marco Moreto and Paolo will present their approach soon. So maybe,
I know that the Vespucci guys, they are doing a wonderful job about that. Really, I think they are pretty much more advanced than we are on the great platform. So I should really say that, and that's what also the costs networking strides out is actually we need to work together, because we are kind of duplicating the efforts right now, and I think we could avoid that and working much more together on this huge task. And I think that the cost will allow that, and that's for good. So
I think that it would be also good for, at some point, the different platforms available try to link themselves through these ontologies and these approaches. Yeah, it could be nice. I mean, it's a double effort, but it could be nice to include it at some point.
I see a question from Claudia, right?
Yes. So before that, Luciano says that thanks. It does not pop easy on Google when you're browsing it, but I guess it's because it's a little bit hidden yet.
It's not published yet, so it's not fully publicly released. So you'll be like some kind of beta testers today as well.
Are you planning to upload maybe on bioRxiv or some kind of?
Yeah, right now we need to. I should say that thanks to this training this week with Amandine, we did a huge amount of work to actually improve the great platform. So I think it has never been as ready to be published. So we definitely it's definitely going to happen very soon.
Really. So we hope we see the birth of Great soon.
The real birth. So right now it's only the pregnancy, you know.
Yeah. Now moving to Claudia's question, this is quite interesting. She says, "Do you have any idea about the representation in the GREAT database based on RNA-seq data coming from table grapes?"
I think it's quite little for table grapes, because at the beginning we really focused on. I mean, we for example excluded like the seedless varieties. I don't even remember why. No, I don't even remember why. But really, it's mostly focused on wine grapes. But but we could actually change that because there is no real explanation where we filter out. Amandine, do you remember anything? Do you recall anything about that?
No, really no. Mm-mm.
It was, I think, a matter of really comparison because table grapes and wine grapes, they are kind of there is some kind of genetic distances between these two groups, but really I don't think it's really a problem. So but feel free, Claudia, to explore that this afternoon or tomorrow. I don't know when your turn will be, but if you are interested in some kind of table grapes in particular, just have a look and check and let us know if you think that we are missing some important varieties or samples.
Yes.
Paula say why to exclude it. I suppose you are talking about table grapes. It was not. We didn't exclude table grapes. We at some point excluded, yes, the seed, the grapes without seeds because we thought it was not really comparable. But that's, yeah, maybe.
I don't know if it fits in the development of grade, but at least for let's say gene co-expression networks. So you do gene co-expression networks, and this has been published in other species. It's recommended not to use overexpression lines or knockout lines because of those cases, things change so much that add too much noise. So at least from the gene co-expression network approach, it's recommended not to include mutants or transgenics that overexpress a gene. So that is something that could be also considered for this.
Yes. Okay. I guess I addressed Claudia's question. Thank you, Claudia. Okay. Tahir is saying something.
I was going to ask you when would be okay for you to do a short break?
Right now, why not?
Yeah. Okay.
That was actually my hidden plan.
Good. So let's do maybe a 10-minute break so everybody can have a coffee.
Sure.
And we come back?
Yeah, in 10 minutes, yes.
Perfect. Thanks, Camille. It has been really, really interesting.
So far, we'll see.
Okay, yes. So to everyone,
Camille, can you? I'm sorry, just 1 minute. Can you? Would you like to comment on my question?
It's about logging for the GRADE platform. Yes, I'm going. You'll see the link to access the GRADE platform later on. You'll also get the email address. So if you want to create your own account, but for the trainees, this afternoon and tomorrow afternoon, you will get a training account. So and we'll give them to you personally. So one trainee each will have his or her own logging account, but we'll give that this afternoon.
Anyway, I'm putting in the chat what Amandine answered to Luciano, but I don't know if it works for all of them.
No, unfortunately, if everybody is logging using user one, it will crash maybe at some point.
It can work, Camille, because it's with same user. This is different.
Oh yes, okay.
No problem.
Okay.
We can try.
They can discover a great if they want.
Yeah, I mean, you will be able to see it later, so don't worry.
Yeah, you'll have plenty of time, and don't hesitate to request for your own account so you can explore it anytime.
Okay, thank you.
Okay, then. So see you again in 10 minutes.
Yes, see you. Shall we move on?
Hi, Camille.
Hey.
In 2 minutes is okay?
Yeah, sure.
Okay.
No problem.
Camille? I don't know if you answer about the question of Paola or no. About
Not yet. I'm going to
Okay.
wait for the other
Yes.
to be back,
Okay.
and I will.
Okay.
Sure.
Hi, Camille.
Hi. Is it that cold in Spain?
It's super cold, yeah. I have to.
Super cold, like what?
No, super windy actually, and the wind is not so great. No, here's the problem. There is a lot of wind in Zaragoza.
Okay.
And also, like as a cafeteria, we cannot be a lot of people. And to make sure that there is not a lot of people in the cafeteria, the waiters are very, very, very slow. Like this, to give the time to people to finish the coffee, and the turnover is not too bad. But it took them like 10 minutes or more to serve one person before me.
Oh, wow.
It's snowing.
We had snow like 2 days ago in Alsace. -5 degrees.
Yeah, that's why I cannot complain. But
winter is coming.
no, because we are still at 16 or so in the afternoon. But when we do have wind, it's terrible. I prefer cold than wind.
Well, that said, shall we move on? Yeah. So I just
they're agreeing with me.
Before keep going, I'd like to address Paula's question. So she said, could it be possible to include table grapes into the database and then filter for the comparison of just that group to avoid mentioned problem that occurs? So Paula, really what you should do during the training session is have a look and see if you can find some table grapes. and then you can come back to us, and we can actually check with Amandine if it's if there is really a problem of comparison between table and wine grapes. And if not, I guess we can just include them all the time. And if there is any, we could maybe think of developing a great platform dedicated to table grapes. That's possible because, as I said, it's great is meant to be fair, so we can actually use it and develop it for and apply it to any other data set. So we could, or if you want, even you could, or with the help of bioinformatician around you, develop your table grade. And it's
that's a good one, table grade.
and so the other question would be about the hybrid. Free hybrid is definitely a problem because we have definitely this genetic distances between the other Vitis species that are within the genome of the hybrid with the Vitis vinifera genome. So that's a problem. You can probably find in the database already some hybrids, maybe you know Regent or Bronner. So these are some resistant varieties that are hybrids. So you can find some in there because most of their genome is actually Vitis vinifera. It's not half vinifera, half another Vitis species. So that's why we kept them. But really, F1 hybrids, for example, it's hard to deal with them in the grade database. But we can, as I said, for the table grade, we can have hybrid grade or any kind of grade we want. But then we'll have to think about which reference genome to use to actually align the reads against.
Yeah, you have to think that it's already difficult or complex between cultivars. There are so many structural variation between cultivars, you know, like completely rearrange of chromosome regions and gene duplications that are in one cultivar and not in the others. So it's already difficult within the same species. So you can imagine how difficult it would be with hybrids. But definitely this has to extend to that at some point, right? David is also asking, yeah, in this case, if the rootstock Richter 110 is in the database.
Rootstocks. Rootstocks, most of the time, from what I know, I'm not an expert in rootstocks, but most of the time these are either hybrids or completely other Vitis species than Vitis vinifera. So the problem is the same. But I'm very confident in the really near future, because with all the genomes that are sequenced and assembled right now from other Vitis species. I know there is a Vitis riparia already, there is Vitis rupestris, and definitely we could use this assembled genome as reference to align all the RNA-seq available for these specific species.
Yeah.
And really, philosophy of GREAT is you can apply it on any species, any genome that you want. So we would be very happy if GREAT actually multiplies itself with other databases.
Yeah.
So really,
Well, in the case of Richter 110, I believe it's a cross between Berlandieri and Riparia.
okay.
So
I think so too. But yeah, so there is no rootstock that are Vitis vinifera.
No, I mean that's the whole point, I guess.
Yeah, totally. So if we knew that there is some phylloxera resistance in vinifera.
We'll see one time, maybe if we breed some resistant vinifera varieties. We'll see. Who knows?
Yeah.
I'd love to see that in the future. But well, that's not going to be my job, obviously. I leave that to others. Okay, shall we move on, or do you have any other
Let's
questions?
continue. There's no more questions so far.
Okay, good. But there are plenty, so it's very interesting to have this kind of discussions. Really, I enjoy it.
Yeah, well, it's just a comment. Claudia is saying the inclusion of table grapes in GREAT should be very useful for sure.
As I said, check during the training session and see what's in there. Okay, so
And also there are we study are kind of new, so that's another thing. So that you cannot include. You have some delays between the moment that the data are published and you can include them, and you have also some work and some priority to do, and not so many people doing that job. So that's also a factor that have to be taken into consideration.
Yeah, I'm not saying that we are going to actually make all these other great platform dedicated to other species, because it's very difficult. Actually, the time is actually to create the database on the regular basis, and it's already very hard to keep going with the Vitis vinifera. So I hope Lauriane is not listening, because if I tell her that she's going to take care of other Vitis species, she's
will
just
kill
going
you.
to leave. So, but we are really happy to help if anybody wants to apply GREAT on any other Vitis species or any species.
Yeah, actually, Andres from Australia, he's saying that in Adelaide they are sequencing and assembling the Berlandieri genome, and that Ripara is already accessible. So yeah, in the future it would be nice to have these references for rootstocks.
Yeah, definitely. But still, hybrids, I don't know how we can deal with that really. But we'll see. We'll have to be creative.
Yeah.
Okay, so let's go back to the actual the GREAT that is out there right now. So I spent some time to explain the database that is behind the GREAT tool right now for Vitis vinifera. I'd like to spend very little time on the workflow for RNA-seq data analysis, because Ricardo did a wonderful job for me yesterday. He introduced the whole thing, so that's perfect. And actually, it's a very similar workflow compared to what he presented yesterday. The workflow is the whole pipeline is written in SnakeMake, which allows very high reproducibility of the pipeline, and to actually keep track of all the stages and the files that were generated at each step. So which are the steps? So first we perform quality control using FastQC. So you've seen that yesterday. We align the reads against the current version of the reference genome, so PN12X-V2, which is available at that link. We are using the STAR software to do that. We perform some quality filtering and check at that step. So if at every step, actually, we perform quality checks and see if every sample reach the threshold that we set up. But it's about 95% of the samples that actually made through the whole process. So we are filtering out very little on the quality criteria. So we use the star alignment, and then we perform gene quantification with the feature count. So this the counting of the reads is performed using the latest gene annotation, which is the VCOS v3, which is available using that link. And then the last step is actually creating the final files, and especially perform normalization for the read counts before using these to be shown and explorable in the web application. I can see lots of going on in the chat. It's okay. Yeah, well. So I keep going. Do you know that you can interrupt me anytime, guys?
No, sorry, it's because I have to. It's because people can only talk to us and they cannot talk to each other, and there is some debates on the hybrids, and I have to relay the messages.
Okay, but you know, guys, that I won't take the whole time until noon, so we can save time between 11:30 and noon to have a discussion on that. Really,
sure.
an open discussion.
Please go ahead, and we will have a special time for questions and open discussion. So yes.
Or
please.
during the e-lunch.
Yeah, also.
Yeah. Good. Okay. So I'll move on with the normalization, because I think it's yeah, well, it's an important point, because it's really the heart of GREAT is to get numbers representing gene expression that can be and these numbers needs to be comparable with everything, for every genes and every sample that's in the GREAT tool and application. So there's going to be a little math, but I hope it's going to be understandable. So as I said, we don't have a proper name for this naming for this normalization. So right now we call it RPKM-like normalization, because the philosophy behind that is the same than RPKM, meaning that you should produce a value that makes everything comparable with each other. Except that we are not using the number of reads mapped to perform the normalization. We are actually using the normalization that is included into the DESeq2 R package, and it's based on a mean of ratios method. So I'm going to briefly explain that to you. So let's say we have two samples, sample A and sample B for 5 genes, but obviously we have 30,000 genes, so I'm not going to show the whole thing. So this is just an example. And we have read counts, so coming from feature count, so the previous step. So for example, we counted 1,489 reads for the EF2A gene in sample A, and 906 reads for EF2A in sample B. And so the question now is how can we make that comparable? So for this gene across all samples, but also across all the genes. So the first step is actually to build like what they call a pseudo reference sample. So using so they multiply the number of reads and they take the square root of that. And so they calculate what they say in the DESeq2 package, what they call a reference sample. And so you get a new numbers that is actually used as a reference to perform the normalization. So you do that across all the samples. So here there's only 2 shown, but in the GREAT database, we performed that on just about 2,000 samples that we have. And we have to redo the normalization every time we add new samples. Then when this pseudo-reference sample is built, you calculate a ratio, so the ratio of the read count from sample A compared to this new number, and so you get a ratio, so 1.28 here, and you do the same ratio for sample B compared to the reference, so you use this number of reads divided by the pseudo-reference sample, and you get a ratio of 0.78. And you do that for all the genes. And so you get the ratios for all the genes for every samples under study. And at the end, you calculate the median for all these ratios. So for the sample A, the median is 1.3. And for sample B, the median is 0.77. This ratio is then used to perform the actual the normalization, really. So it's really this normalization factor that is used to normalize according to the size of the library, let's say. And so so you take every read counts and you divide by this normalization factor for the sample. So here, this read count divided by the normalization factor calculated for sample A, and you do that for all the genes for sample A, and you do the same with the normalization factor that you calculated for sample B, and you do that for all the genes. And this way, you end up with a numbers. So these are not really read counts anymore. They are normalized read counts, let's say, but it makes this number comparable across different samples. So you can actually now compare expression, and you can say for the gene ABCD1, the expression is higher in sample A than in sample B. So it's pretty similar, let's say. If you really want to say something, you move on to gene move 10, and you can say definitely that expression in sample B is greater than in sample A. I can see a lot of stuff going on. Okay, in the chat.
It's just that we just reconfigured the chat so the attendees can talk between themselves. I mean,
I see. I know that the math, they don't like that, so they want to chat with.
But yeah, there are no questions right now, so don't worry.
Okay, no, I'm not worrying. I know that you are taking good care of that. Anyway, so that's the first step of the normalization using this DESeq2 normalization method. So it makes expression for a given gene comparable across samples. But definitely at that step, you cannot compare these numbers between different genes. So I'm not able to tell right now that the MEFB gene in sample A is more expressed than the ABCD1 gene in the same sample. So I'm not. Right now it's not normalized, so I can compare the genes with each other. So I have to move on to the second step of the normalization. If I can change, yes. And this looks pretty much like what you may know about our PKM calculation, and we are using here gene size in KB to actually normalize this already normalized read count, let's say. And so to make the expression values comparable between genes. So let's say the genes have these given sizes, and you use the previous normalization read counts that we just calculated, and you divide that by the gene size in KB, and you end up with new normalized value, which is actually the RPKM-like value. And so you do that for all the genes in every sample. And at the end of the process, you get numbers that are comparable between samples and also between genes. And so this is really, as I said, the heart of GREAT, because we really wanted to have all the values, the expression levels, the expression values comparable with each other. But it's definitely not the same philosophy than when we are performing a different expression analysis, where actually you want only to compare the expression of one gene in different conditions. I hope this is clear. If you don't remember how the calculation is performed, it's not critical, but it's just to give you a bit of a detail about how we are doing things. So this was about so the workflow. So as I said, it's very there's nothing fancy about this this workflow. It's a classic way of doing things. You can actually access it through the GitHub. I don't think we put that. Did we put that somewhere? Yes, actually, I forgot to say that. So you can access and reuse the whole workflow using this GitLab address, and so you can apply it on any RNA-seq data that you want, as soon as you have access to a Linux server that can handle this kind of RNA-seq data. Okay, so now I'm going to present you the web application for actually visualizing the expression data. For most of you, this is the only part that you are going to explore and to watch really, and to use at the end. So just so you know, this visualization, this web app is based on 2 R scripts. It's developed in R Shiny, so it's not like a web page, let's say. It's every time you log in, you actually launch on the server. You launch you are launching an R software somewhere, which actually performs some calculations, not all of them, but sometimes when you see it gets slow, it's because behind there's a lot of calculation going on, and so it's because you don't see it, but you are using R when you are on the GREAT web application. So that's what you have to keep in mind. What you see is the results of calculation of R. So yes.
There is a question that maybe will fit here from YouTube. It's raised by Joseph Tran. I mean, somehow I already answered him, but I think it's a quite interesting question. He's asking, what do you mean by gene size? If it includes the introns, and why not normalizing on the CDS size? Yes.
And I answered that I think, but I have to verify. This is the sum of basis of all exon of a gene.
Yes, it's actually for the to make things easy, but it's probably not precise enough. I should have said transcript size.
Yes,
So it's really based on the.
However, I must say that depending on the annotation file, on the GFF file, sometimes you have Gene as the transcript, and a problem in the annotation of the annotation file.
Yes.
Within the annotation, right? Because
yes.
depending on the version, depending on the species, for instance, I also work with tomato, and I've seen that what they call gene on the annotation file is not the same as what in grape we call gene. And this is a problem of the standardization.
Yeah, exactly.
Yeah, so sometimes gene, the concept gene on annotation file only includes exons, and in some others includes the introns as well. So please be careful for everybody, for the observers and everybody working on these. Make sure what each of the features is being mentioned or represented in the annotation file.
But definitely here the normalization is performed using the transcript size. So it's based on the UTRs, everything that is transcribed actually. So UTRs and exons. So not only the coding, but transcript. If I mean, it also depends on how good the annotation file is, because sometimes UTRs are not really well annotated. We know about that, but still, it's supposed to use the transcript size.
Yes, and I verified in the script, and yes, it's the sum of all bases of all exons.
Exons, yeah. So transcript size. Good. Good questions. So okay, so finally the addresses and that everybody requested. So the grade is available online. I know it's a bit hidden still because it has not been published yet, but anyone can see it using this link. So it's grade.colmar.inrai.fr. If you click on this link, you'll end up on the login pages. So you'll need a login. You'll need an account to actually use Grade. So if you want to request an account, just write an email to contact grade.colmar@inrai.fr. So Amandine, me, and Lauriane will get this email, and we can create an account for you, even if the publication is not out there already. But you can start using the Grade application. And if you do, please feel free to actually send us some feedback. So you'll notice it's a tool that is still under development, and so we are really appreciating that when users are giving some feedbacks, comments, suggestions, it helps to actually improve the Grade tool and to make it even greater, if we can say, even better. And so yes. So do not hesitate to give us some feedback. Thanks in advance. So how does it look? So as soon as you're signing in, as soon as you're logged in, so you can see these pages. So this is the home page. So you can it actually describes the tabs that you have up there, and to give you a little bit of explanation of what you can actually do using these different tabs. And I will go through these tabs with you right now. So first, there is you can access the Grade database. So all the Excel spreadsheet I've talked to you about. So the curated one that Lauriane did, and built, you can access it all. You can download the entire GREAT database. You can explore it on your own computer. You can filter the database. You can actually look into it by typing there some anything you want. So, for example, if you want to see if your dataset, the dataset that you published 2 years ago is in the GREAT database, just have a look. You can have it right here. But really, this is very important for us to build GREAT as a fair tool. So we really want everything to be accessible to anyone who requests an account, but it's we give an account to anybody who is actually asking. So you can download the whole curated database. Then you can have a look at the quality control table. So we give you access to all the metrics that we keep track of, the number of reads, the number of reads aligned after filtering, the number of reads or the percentage of reads that are assigned to an annotated gene. You can actually check for every all the 2,000 samples that are in the GREAT database. You can actually have access to all the quality criteria that we make. But then maybe a tab that you'll explore a lot, especially starting this afternoon, is the tab that is called Gene Expression Visualization. So in this tab, you can actually draw heat maps to visualize and to look into a gene expression profile across all the samples in the GREAT database. So Amandine created different kinds of filtering here. So you can choose type of treatment. So if you're looking into a biotic treatment, and all of this, you don't have to type. You can select and you can choose for among all the different treatment tags that are available. So you can just click and select the one you want. For the treatment nature, you can select either Plasmopara viticola or any other biotic treatment that is available in the GREAT database. You can then select the variety you're interested in. As soon as your variety of interest has suffered from a biotic stress with the Plasmopara viticola, actually the choices that you can make are updated at every step in the process. So you'll see that this afternoon. You can also select the organ you're interested to look at. So most of the time, the treatment types are applied on leaves, but not always. And then you can also filter if you want on a specific bio project, or you can just choose to select them all and have a look at all the bio projects that are actually fitting with this criteria. So biotic stress, Plasmopara viticola, Pinot Noir, and leaves. Then you can, so this is about which samples with conditions you want to explore. And then you can also select the genes that you want to look at. So either you choose the method of selection based on the gene lists. So you have to use the gene IDs from the VCOS v3, but we'll see that you may, we are still working on that, but we'll see this afternoon how we can translate, let's say, the gene IDs in different version of gene notation. But anyway, if you choose to work on a list of genes, you'll have to give the list of the genes separated by a comma. And so all these genes will be used to draw the heat map. And then you can choose either to view the expression value either in these RPKM-like normalization values that I just explained to you, or you can also choose to look at the log of these values, log +1 because of the 0, which causes problem. But so you can have access to the normalized values or the log. And usually we use the log one because it actually helps to how do I say? Yeah, the values are more centered and are smaller, and it's easy to actually compare everything. But I'll let you explore that by yourself. So either you you choose a list of genes to explore, or you can also give some chromosome coordinates to look at all the genes that are annotated within a given genomic region of the genome. So you can choose to look at what's going on chromosome 1, starting at that position and ending at this one. You can also choose, if you want, the strand that you want to look at, plus, minus, or both. So if you want to look at both, just leave the star. That is the default here. And then you can, again, choose RPKM or the log and launch the analysis. And so this way, after filling all these different boxes, you end up with a nice, what I hope to be a nice heat map. So you see here, probably this heat map was performed on given genomic region, probably chromosome 2, as I can see here. So we have like, I don't know, 30 genes or so that are represented here. And on the other axis, you have all the samples that you selected, that were selected for building this heat map. So sometimes if you select too many samples, or you can select actually all the 2,000 samples, it will work. I can already see Amandine struggling with with that, but it will work, except that you probably won't be able to read the labels here. That's why Amandine worked on these little pop-ups here that you can see in gray. So if you navigate with your mouse on the heat map, you have access to different information. So the name of the gene that you're looking at, some information about the sample, and the value, so the expression value that you are looking at. So this is probably a heat map that were generated in logs. So the log value of expression is 7.6. Yeah. So you can already see on this heat map that some genes so when the genes are highly expressed, it's reddish. When it's not so expressed, it's mostly blue. So you can see here that some genes are not expressed at all. So this is the case of the gene here. Some are expressed in some conditions here, but not in others. Some are expressed a lot for all these samples. And it gives you information on the expression profiles of your genes of interest. Then the next tab is actually the table that is used to build the previous heat maps that I just presented you. You can download all the table where we store the expression values. So you can access the whole thing at every step of the analysis. You can really get everything. So if you, for example, after your first data mining, you've seen that some genes are very interesting. You want maybe to draw your own figures, your own plots, using whatever software you want, Excel, R, anything. You just export the table, and you can do whatever you want with the expression values. So that's also good thing to know.
Just a short question. I'm not sure if you mention it. So do you do the filtering of lowly expressed genes, or you include them no matter how many?
We do not filter out any genes at any time. We keep everything.
You keep everything.
So if a gene is not expressed at all in any of the 2,000 samples, it is there.
Okay.
So yeah, as soon as it is annotated in the VCOS v3, it's there.
And the PCOST annotation has many more genes compared to the previous versions, because it also includes a long non-coding RNAs and microRNAs. And are those also represented here, or they are already filtered out in the, let's say, in the matrix of the GREAT database?
I'm not sure. I think Amandine should confirm that, or we can confirm that later on, but I'm not sure we filtered out anything. But Amandine?
No, I use the raw GFF3.
Okay.
I keep all the genes, yes.
Okay.
So as soon as there's an exon annotated for something, it's used for the counting. I assume. Yeah.
Just a clarification here. So the V2A is a merge between the V1 and the version that is on the NCBI. And all of these non-coding, they are coming from the NCBI because they take them into account.
But it's, I mean, it can be useful actually to have a look at them. If you're interested in non-coding or stuff. Amandine, yes.
Yes, some ncRNA have no exon, so in the GFF3, so I don't take them, into account, for the analysis.
There is some exon, but there is no CDS.
Maybe, but I just use the exon part, so. But
as soon as there is an exon that is annotated, usually you're taking it. It's not CDS. It's really exon.
Exon, so they should be there. So actually, the difference between the non-coding and the normal genes is that there is no coding. There is no CDS for the non-coding. So, but if you have taken the exon, they should be there.
But yes, just to mention that they are kind of. Not the majority of the genes. They are kind of
it's a very small proportion of
a small proportion. But yeah, it's yes, we really rely on what's in the GFF3 file, definitely.
And this makes me raise the question about, because when you download the SRA datasets, if you search for them sometimes, because I have encountered this problem that while downloading RNA-seq datasets, many times I come up downloading without knowing, but then I have to remove microRNA-seq or small RNA-seq datasets. So in this case, do you filter them also when you're downloading them from SRA?
Yes, we are not using small RNA samples here, not at all. Usually, Lauriane is able to catch them before we download because she's really getting into the publication, and so. But it's hard work. Sometimes we ended up downloading this kind of data sets, but with the length of the reads, we notice immediately that these are
for sure. The point is that going back again to the metadata issue, sometimes microRNA or small RNA experiments are annotated as normal RNA-seq. Yeah.
Most of the time, Lauriane is actually catching these because she goes through the material and methods section of the publication, and she can actually retrieve that the library protocol was a small RNA, blah, blah, blah. But yes, you're right. And it's again, we cannot say that enough. We need to be as precise as possible when we submit metadata associated to any data we are submitting. Definitely.
Yeah. Thanks.
Good point. Good. So let's move on. Another tab that Amandine actually developed recently is the gene expression clustering. So only a subset of analysis. So this is not sample focused anymore. This is a more experiment focus here. So, for example, I think there is probably 20 or 25 experiments that are included in this clustering section. So, for example, she retrieved all the Blanco, all the experiment from this author, and she allowed to perform clustering analysis. So what does it mean? So for example, let's move on to the next one. Yes. So in this experiment, it's probably time scale analysis or developmental kinetics or something, looking at gene expression across different developmental stages, or looking at expression, for example, when you apply a treatment, 1 day after the treatment, 2 days, 3 days, 1 week, and so on. So using this kind of data sets, using this kind of experiments, you can actually look into the genes that are behaving, let's say, in terms of expression in the same way. So you can build clusters. So for example, here, in this tab, using this experiment, you can see in cluster 1 the genes that are not very expressed for the 2 first time points, but then the genes are getting expressed starting at the 3rd, 4th, 5th, and 6th time point of the experiment. The cluster 2 is the opposite, is for the genes that are mostly expressed in the first two time points of the experiments, and then the expression decreases. Cluster 3, I think you got the idea here, is for the genes that are specifically expressed for time point 3 and 4. And so then you can, using this clustering, you can actually extract the genes that are specifically clustered together, and explore them a little bit more in terms of their function, in terms of their expression in other conditions than the one in this experiment. And so this can be useful for some biological questions, for example. And we'll have a case study using this clustering tab this afternoon and tomorrow. then another new tab here that Amandine developed recently is the differential gene expression analysis. So I know I told you GREAT is not meant to perform different gene expression analysis, but the thing is we had some requests from biologists saying, okay, there are out there a lot of different publications doing differential expression analysis, but I'm never sure that they use the same workflow to analyze their data. So I'm not sure if I can really compare their lists of differential expressed genes. Actually, we for the experiments where differential expression analysis were performed, we did that too, but using for all the experiment listed here, we use the exact same protocol for differential expression analysis. So we didn't use the RPKM like a normalization method, just like I presented you yesterday. We used a dedicated workflow for differential gene expression analysis, and Amandine can confirm, but the workflow is also available on the GitLab.
Yes, it's the
repository.
ASCO. Yes, it's the ASCO-R pipeline.
Yeah.
It works with CPM as a normalization part.
But anyway, the goal here is really to make different differential expression analysis comparable with each other, and to be able to cross the list and to see if some genes can be found differentially expressed within different experiments.
Can I ask a question here?
Sure.
So the CPM method is the one you use for normalization, and for the gene expression, which pipeline you use for the differential gene expression analysis?
Yeah, it's ASCO-R. I can write it in the.
Okay.
And
just as a curiosity, basically.
And the pipeline use EdgeR. You know. So.
And finally, for the clusterization method, which clusterization method you use?
You mean for the previous tab there with these?
Yes.
It's the MFuzz package. You can write that down too. But again, everything is in the GitLab if you want to reuse it. So yeah.
Great. ASCO-R, yeah.
Yes, ASCO-R is it's the name of a workflow actually using EdgeR as a package to identify the differential expressed genes, but ASCO-R allows also to draw and to write reports, whole reports for the analysis. I mean, for quality checks, for PCAs, for example, of different for the different samples, so on and so forth. So it goes beyond only what EdgeR is doing. But so EdgeR is included into the ASCO-R pipeline. But we were thinking of maybe changing that workflow, going back to SAR tools. I think that Amandine, you wanted to use.
Yes, I think about that. I don't know for now, but yes, maybe SAR tools. I don't know.
Because SAR tools is well known, and ASCO-R is more confidential. I mean, it's a colleague of ours in France who developed that, but it's not very well known abroad. So
I haven't heard of it before. Nice to see. There is one question from Joseph and Alberto in the YouTube. The to do with this, so that's why I will ask them. They say, why do you normalize by CPN instead of RPKM?
For differential gene expression analysis, definitely normalizing using gene size, transcript size causes problems, and it's not the philosophy of a differential gene expression analysis is not to actually compare genes with each other, but it's more like comparing the expression of one gene across different conditions. So actually normalizing using the size of the gene or the size of the transcript adds a bias to the analysis. So when we are doing differential gene expression analysis, and that's what Ricardo said yesterday, exactly. We cannot use RPKM
Also, I would like to add that the RPKM method does not include RNA composition. So actually you don't know or you don't ponderate the presence of the different molecules on the complete bag of transcripts.
Yes.
Yeah. And then Joseph is asking maybe to find some COGS. Okay. No. He was asking before about the grade being extended to small RNA-seq data set, but Amandine already answered that.
Okay.
Okay.
Good. Good team. Okay, so so the differential gene expression analysis. So if you run that on GREAT, you will end up with for a given experiment all the different conditions within the experiment will be compared with each other. So we'll have you can access them then to every comparisons possible for a given experiment. So they may not mean anything biologically. I mean, they may be pointless, let's say, for a biological point of view, but it's done automatically, and if you want to have a look, you can. That's how it is. And so you can you have access to the number of genes that are found significantly upregulated, downregulated. You can download the whole table. If you want, you can download the whole report analysis. You've got access to anything. And heat maps will come soon because we need heat maps every step of the way. We love that. So then moving on to the almost the end. So do the functional gene annotation tab, and here, and I'm going to do it also on the next slide. We cannot thank Jérôme enough because it's thanks to him and thanks to his work, Jérôme, that we could actually add this new tab to the GREAT app, and I think it really helps with the data mining process to actually have access to the function, the putative function of the genes we are looking at. So many thanks, Jérôme, for that. So you can actually retrieve or look at the function of the genes you're interested in, either by looking at the list of genes of interest, or either by exploring a region, a genomic region that you're interested in. Hang on, just I need one.
Sorry, I think your microphone got mute.
Yes, I did that. It's because it got very noisy in my house right now. That's the problem when you're working at home. Anyway. Yeah, So I've got birds, and they are getting very noisy at that time of the morning. Sorry for that. So you can actually access the function of your gene, either using the gene list or using the genomic coordinates of your region of interest. So again, you mentioned the chromosome, start, and coordinates the strand you're interested in, and you access the function of the genes that are annotated within the region. So again, thank you, Jérôme. And so using this filtering, so you have access to the information on the functional annotation. So, for example, so this gene that is annotated in the V3 because V3 version. So that's its gene ID. But what is very, very useful also with this table is that you can access to its ID in the other version of gene annotation. So, for example, for the V1, its ID was 1. For the V2, its ID was this one, and in the RefSeq gene annotation, that was its name. So this is also a great table to actually convert, if you'd like, between different versions for the IDs of the genes. And so you can then see the functional annotation of the gene. Sometimes there is none, so that's because no homology was found with other functionally annotated genes. You can see the best match with Arabidopsis. You can access the gene ontology for this given gene, or even the plant ontology if you want. So this completes nicely what we can do as a data mining within the GREAT application. And finally, so the tool ends with the user guide. So Amandine is very willing to keep a user guide as up to date as possible, in order to explain you as much as possible and to guide you through the GREAT app. But as I said, if at some point you see that anything is complicated or not obvious or not well explained, please email us back to tell us, and we can add some stuff in the user guide. So it's really meant to help the user to navigate into the GREAT app. So as I said, also GREAT is not it's not even born yet. So of course it's regularly updated. So as I said, there are new tabs that were added recently. The functional annotation tab, the clustering also is new, and so it's still an ongoing process, and we are and Amandine is working hard to make a lot of improvements to make the GREAT app as intuitive as possible. As I said, we try to add new data sets every 6 months. So Lauriane is going back to the SRA database every 6 months to retrieve new data sets to add to the GREAT tool. There is definitely a publication that is in progress and in preparation. We all hope to get that out as soon as possible. Also, you've heard that there will be a new version of the reference genome that will be available. And as we are part of as we actually did, yes, we improved the new reference, this new reference genome. We also want to use it to improve great and to use this new reference genome to make a great run on the new annotation also that will be available, but it's not done yet, and it won't be available before the release of the new reference genome. But definitely the new version of GREAT will come along with the new reference genome. We are thinking of, I don't really know how we are going to do. Maybe we were thinking of keeping the two versions, the one on the 12xv2 reference genome and the new one, at least for a moment, so people can are not lost by using directly the new reference genome. We'll see how we'll deal with that.
Camille?
Yes.
Maybe would be a good time now to mention a little bit of what has been raised in the different core group meetings and in the different meetings of the cost action regarding actually the new version of the annotation. I know that actually many people has written us during these days asking on which are supposed to be the most correct or the annotation versions that should be used from now on. The problem is that every time a new genome accession is available, a new annotation. So this new genome accession that you are improving with the PacBio sequence probably will come with a fourth new version, right? So new codes, new IDs, and new problems for the correspondence to the other versions. So yeah, I think this is something that unfortunately on a crop species consortium, it's difficult. Of course, in Arabidopsis, for instance, you always have the same ID, and you have so far 10 different versions of genome accessions, and you still conserve the first annotation version.
Yes, it's a tricky, it's a tricky part of the genomics on Grapevine is definitely dealing with genome assemblies is kind of manageable, but the gene annotation version is just a nightmare. So yes, I understand the trouble because I actually am dealing with them on an everyday basis. So yes, I know that having different gene IDs is a problem. I think that Jérôme has done a pretty good job at trying to actually link the different IDs across the different gene annotation version. So that's very, very useful and very good. But unfortunately, with a new genome assembly comes a new gene annotation, of course, because we are assembling parts that were not present. We are giving sequences for parts that were missing in the previous annotation. So we'll have to have new genes annotated. Maybe we could actually keep the same IDs right now. We are still in the process of annotating this new reference genome, and so producing the annotation. We are not at the stage of the naming right now, but it's definitely in our mind because we are all struggling with that problem. Yes. Jérôme, you want to add something?
Yeah, and a slight difference compared to what has been done before is at that time, you and Nicolas Vitullo, you are also using this annotation as model to find the new model in the new version. So maybe it could be smoother to do the correspondences between between the V3 and the new version. But here it's more delicate, harder than what we have done between the V1, V2, V3, because so far we were working on the exact same sequences with a different order, better mapping of the scaffolds, but that was improving the version. But now you guys are resequencing from the scratch, so it's even more difficult. But we do have the advantage to use this version to do the new annotation.
Also, we have more data sets. I mean, we've got all these RNA-seq data sets that we didn't have previously for.
It will be better for sure.
So for sure, it will. I mean, I know it's new, and we'll have to adapt again to something new. But it's, I think, in my opinion, definitely for the best. At least we are working that way with Jérôme, with the group of Mario, with the group of Daniela, and so it's really a collaboration within that actually started within this cost action, and we are really wanted to do our best here to make life easier for anyone who is using the reference genome and the gene annotation. So we have that in mind for sure. Then there's the other problem. I mean, this discussion is focused on the reference genome, but there's a lot of new genome assemblies out there, and we are also trying to discuss about a common gene annotation process for the whole grape genome assemblies. We are actually discussing that with Doreen Ware from the Cold Spring Harbor, because the idea is eventually to be able to build a pan genome for grapevine, for grape. And so that means we will need to be able to compare gene annotation across all kinds of different genome assemblies. So that's another layer of complexity compared to only the reference genome. So we have to work on every side of the problem on the reference genome, but also across the different genome assemblies. It's the main problem I can see right now in the grape genomics area.
Yeah, my question is also focused to the fact that if we even improve the version that you are producing with new sequencing technologies, and we come up with a new assembly, and we would change to a fifth annotation. I mean, I don't want to give this problem to you in the sense that you don't have to answer. It's just an open question to all of us, actually. We really need to be to take care of these, because I know that depending on the region of the world, there are people still using the 8X.
Yes, I know.
There are people using the B1 annotation. There are people using the BCOS. There are people using both of them. I think that we should standardize this as well.
We need to be more, I would say that, visible, or at least to have a hub somewhere where everyone working on grapevine genomics knows that he or she can find the up-to-date version of anything. I think, because right now, if you go on the NCBI, there isn't the current genome assembly is not there. If you go to EMBL, it's not there either. It's at URGI in France. It's not very well. Yes, the communication is not good right now. But
maybe it's worth mentioning that right now you can have the 3 versions available at the grapegenomics.com website from Dario Cantu Lab. So there you can upload the 3 genome browsers, and you can have the models of each one of the annotation versions, one next to the other, so that that can help some of you to compare the models and so on. But also, as some of the trainees have asked in the chat, you can find right now the conversion table from B1 to BCOS.
I'm not sure about that.
What do you mean?
But it's on the URGI.
It's on the browser. Actually, if you go on the Gbrowse, and you can actually see all the different versions, because I've worked with them on that part.
But I have them on an Excel file actually, also on the GitHub that I gave yesterday. I will put the link again. There is some version on the URGI maybe of the Excel file.
Yeah, but it's the 3.3 version. The one that you have in the GitHub is the 3.26, I believe.
Yeah, because we are doing update every once in a while. So this is a work in progress. We are not working too much because like we will be moving on the new version from Camille. So right now it's a little bit stuck. That's also why we are not advertising it a lot. Either not to confuse. But that's so we are constantly doing some updates. Actually, the difference between the V25 and the V26, Tom, if you remember, is because one of the genes that you were studying with Luis, you found something wrong in that version. So I did, and we did the modification in the GFF and the Excel tab. And the latest version they are in the GitHub.
Yeah.
Because the one that you may or may not find in the at the URGI should on V20 or something.
Yeah, maybe you can send the link.
I will
have in the chat and also in the YouTube. On the YouTube, yeah.
What I wanted to say as well is that at some point, the improvement of the genome assemblies, they reach a point where the improvement that after one more step is not huge, right? So most of the what we have for as information right now is correct and is not going to change, and most of the sequence will remain the same. So it's every time we are improving the assembly doesn't mean that every time we change the sequence completely. So we really have to think how we can keep gene IDs among the next version of gene annotation, because definitely the sequence won't change that much anymore in the future. Even now, it's not changing that much compared to the 12xv2 sequence, definitely. So it's not a chaos every time. So just to make things just a bit less bad than it looks. But yes, it's definitely when we are doing RNA-seq experiment relying on gene annotation, it's very important. What we have in mind with Amandine is at some point maybe allow the community to create their genes using Web Apollo tools. So it would allow people who are actually expert in some, for example, gene families, transcription factors families, to actually annotate or to make modifications where they think there's a mistake. And so it could actually help improving gene annotation, yes, and help the GRAPE community to end up with something that is really curated. Because I'm not an expert in every gene family. I only know a few of them. I can correct some of them and be confident, but not all of them. So I think it's really a team effort, but we'll have to think about more involving the community more, maybe, for this matter of gene annotation.
Yeah, definitely.
Mm-hmm. Well, I'm almost done anyway. So,
But we have plenty of questions now.
Yeah,
sorry?
we have questions for the great platform.
But maybe I can just finish because it's only acknowledgements, and then we can move on
Sure.
with questions. Let's do that.
Sorry for so many interruptions. Sorry.
No, it's okay. That's the whole fun of it. So yeah, I just wanted to acknowledge a whole bunch of people. Amandine, I don't know how to thank her as much as I wanted, but really, it's I'm speaking about great, but it's her baby, really. So Amandine, thank you for the effort. Lauriane as well, Lauriane Renaud, who is doing a wonderful job at creating the database. It's a very frustrating job because you cannot really see it. The whole database, the whole great tool is built on her work. So really thank her. I want to thank her a lot. All the other people who were involved. So Jérôme, I thank you for the functional annotation of the genes. You can access, so I put here the links again to access to the great web application. You can request an account using this email address. So please feel free to write us. Just a quick introduction for the training this afternoon. So we'll be working on 3 case studies. One to compare two differential gene expression analysis. Another one to study a quantitative trait locus. And then if we've got time, explore a time series for a given experiment. For the trainees, I just want to stress that it would be better if you use Firefox as a web browser. Chrome is working, but we've been struggling with some little problems, but could work, but we would like Firefox better. I hope that you have access to any spreadsheet software, Excel-like or any others, but it will be needed for the training this afternoon. If you've got problems with that, we can deal with that during the e-lunch. And then I'd like to make acknowledgement to the Metadata community and this great team, Tom, Jérôme, Anne-Marie, Mario, and Françoise, all the support team of Sequentia. They are doing a wonderful job because I think it's really working well, and it's really nice to actually keep going, even if the virus is keeping us at home. I'd like to thank the trainees for their questions, their interest, and helping us to keep improving our tool, and the observers as well. And please, the observers, feel free to give us some feedbacks as well. So thank you all for your attention, and let's discuss anything you want. So there are
Thank you so much, Camille. Really, really interesting. And I think that both trainees and observers are were quite interested in this talk, and they are expecting for this afternoon and tomorrow afternoon training sessions.
Mm-hmm.
We do have some questions. Let me go first to the YouTube streaming. There is one that says, "Hi Camille, as you mentioned that you are not filtering genes, but there is the possibility that there could be discordance in the result later, as particular genes might have lower read quality, lower expression, and map to multiple regions."
Mm-hmm. That's true. That's correct. It's a tricky one, actually. I'm much. Maybe at some point we could, I don't know, but on which basis are we supposed to filter genes out? That's my question. It's if you filter a gene because you can never see any reads aligned to it, it's not going. This gene is not going to add any bias to the analysis because there is no multiple map reads or this kind of thing. So the filtering these out would be easy, but there's not much of a meaning to filter them out. Yes, I guess at some point we really rely on the quality of gene annotation, and we well.
No, it's true. Then we have another question saying, regarding multiple gene families or transcript isoforms, I guess he's referring to splicing variants. Will GREAT include transcript level pipeline analysis?
At this point, no. The thing is, it adds a lot of complexity to the analysis if you add all the layers of alternative splicing. So right now I don't know if we choose one isoform, or anyway in the VCOS-B3 there is not a lot of mention of different isoforms anyway, or alternative splicing.
Yeah, only has.
Yes, only the V2. And I should say working on the VCOS-B3 helps with that matter because we don't have to deal with it somehow. But it's definitely something that we need to have in mind. Amandine, do you want to add?
No, there is isoform in the NCBI version, and that they are in the V3.
No, I will say we don't take it into account the transcripts. We only count reads on exon. And I don't know, all RNA-seq data are not okay to perform transcript analysis. So, you know, if the length of the reads and so on, so.
Yes, for technical reasons, it can be difficult to assess actually. And also, what I wanted to say is that by looking at alternative splicing, we are really starting to touch at the diversity, let's say, of the expression. I mean, the different forms, and here. It's with the GREAT application, it's rather difficult to go into that because we want to compare everything with each other. Alternative splicing may be very specific to a given variety or a given organ or given stage. So it's getting very particular. I mean, a very specific problem, which is not and it's not really the philosophy behind GREAT to go into such detailed stuff. Probably looking if you want to look at precisely at alternative splicing, we will have to use the right, I mean the best genomes. For example, if you want to look at alternative splicing in Cabernet Sauvignon, you'd rather use the Cabernet Sauvignon assembly to look into that. Really, it's probably too much details for the philosophy of GREAT, let's say.
Yeah.
As I said, GREAT is meant to be to help biologists to get some preliminary hints for the genes of interest or the regions they are looking at. It's probably not meant to produce publishable ready results, right? If I may say so. It's more to as data mining tool.
Yeah. Then there is a comment by David also that says, maybe we could recommend a more stable annotation to use, or better we wait for the new annotation.
Right now, I think that the VCOS v3 is very good. So for me, it's I'm very confident in using this one. Of course, when the new genome assembly will be released, we'll have to use the new gene annotation associated to this new genome assembly. But I think it's also the work of the cost to actually help the community to find what are the more reliable version of anything, and to make them accessible easily, maybe through the COST website, or we'll have to still decide on where we want to put this information. But it's an important point, an important deliverable for the COST action.
Yeah.
So no, because I think David was referring to specifically all the 25 iteration of the VCOS v3, why to use v10 or v20 or v26, and I see that you get confused with that. But many of the versions, they are the dot something. They are involving small change in the GFF format file, some error that we have seen in one or two gene model. Also, when we perform manual curation of some families, there is several families that have been very manually annotated there. Tom did some work on the MIB, or what I did on the Matchbox, mostly transcription factors. So no, it's because any annotation should be a work in progress anyway. So you don't really have to worry about a stable or not stable version, you know? They are all as a stable, but every time a little bit more improvement in the manual curation.
There was a question from Sophia about when the GREAT application is going to be public, will it be available for free? Yes, it will be for free. It's we are working in the academic world, and all of the tools that we are developing, it's going to be for free. It's the accounts, the access. We just want to check how the GREAT app is getting used, and that's why we want to the people to access it using a login and a given account, and also to be able to help the people to know what they've been testing, so we can help them the best we can. But it's going to be for free, for sure. No problem with that.
There is a comment on Luciano that says, free makes it even greater.
I agree.
Okay, is there any other question? One thing, we were having problems in giving website addresses in the chat of the YouTube channel, but we are able now. So we finally put the GitHub address in the YouTube.
Good, well done, guys. Thank you.
The thing is that you might have posted several addresses during the whole.
Yeah, that's why these days people could not see the links, but now every time we send an address, we have to send it through Walter, because only the owner of the channel can post addresses.
Okay, I can just go back there. So anybody also on the YouTube stream that can just catch these two addresses if they want. Yeah.
Okay, Filippo is asking for the afternoon training session, where are we going to connect? So we are sending the links for the meetings, just like on Monday afternoon and yesterday afternoon.
Mhm.
Zoom meeting.
We are going to send you also, I don't know who's going to do that, but someone who's got the email address of anyone for this afternoon. We'll have some files to send for the training.
I will send you the list.
Okay, good. It's mostly, it's for the protocol to follow, to guide you through the great application, and also some rescue files in case you get lost somewhere, so you can still keep going with the analysis and everything. So we've prepared some backups just in case. So yes, I'm going to send you that.
Very well. So we are going to keep this plenary session room open for the trainees because during this hour and a half, we will be able to assist you with the error submission in case you had some problems. And we're going to start the training session for GREAT for the first group this afternoon on the link that we have just included in the chat. So please copy that, and you can click on that before 2 p.m. We will keep this room open for talking with all the trainees for any problem that you may have right now. I don't know if there's anybody else who has any other question for Camille or Amandine.
Sorry, I had to left a little. Did you took care of the question in YouTube?
Yeah, I'm taking care of it.
Tom, I have a question for you.
Yes.
I would like to know at what time you prefer to open the afternoon meeting room.
Okay, should we do it, Camille? Would it be okay to do it maybe 15 minutes before 2 p.m.?
Yes, that would be perfect.
Okay.
Okay, so at 1:45 we will open the afternoon meeting room.
Mm-hmm. Yeah.
Good.
Very well. So thank you so much, Camille. It was really, really interesting.
Thank you, Camille.
And
My pleasure. My pleasure.
we will see the privileges and the amazing things that we can do on GREAT this afternoon. Okay. Thank you. Thank you, Amandine, you too, for all the help. So.
Are we going to stop the streaming on YouTube?
Yeah.
Or how? Yeah. Okay.
So
I'll stop it.
Yeah, for the YouTube observers, thank you very much for your presence, for being here this whole plenary session, and see you tomorrow morning for the training session on the Sequentia Air platform. Hope you enjoyed today as well. Bye-bye.
Thank you, everyone. Okay. I'm going to stop my sharing.
Yeah. Okay, so if there is somebody who has had some problems with the upload